Biobank, and today we're very pleased to welcome two speakers who will talk about what I think is a very important topic, maximising opportunities to publish biobanking research. So of course, any field needs to be underpinned by evidence, and preferably that evidence should be published so that it can reach as many different people as possible, and so biobanking is no exception to that. So our two speakers today are Dr. Amanda Rush. So Amanda is a research fellow at the Leeder Centre for Health Policy, Economics and Data at the University of Sydney. Amanda conducted her PhD in biobanking through the University of Sydney several years ago and has been a research fellow at the University of Sydney for, I think, the last couple of years. And Amanda is also joining us in her capacity as a member of the editorial board of the journal Biopreservation and Biobanking. We're also joined as a speaker by Dr. Beth Caruana, who is the Senior Research Scientist here at the New South Wales Health Statewide Biobank. So Beth undertook her PhD at the University of New South Wales and subsequently worked in industry before joining us. And Beth is going to be talking about the Statewide Biobanks experience in terms of publishing biobanking results and other kinds of opportunities. So I think it's really, wonderful to have both of the speakers here today. We look forward to hearing from you both. And I should also recognise that I'm of course joining you from the Statewide Biobank, which is on the land of the Gadigal people of the Eora Nation. And I'd like to acknowledge Indigenous leaders past, present and emerging, and the many contributions that Indigenous people play in society in Australia and in their ongoing connection to country. So thank you again to our speakers. Amanda, if you'd like to share your slides, and we'll start the presentation. We look forward to hearing from you.
Just waiting for it to load, Jenny.
We can see the slides.
Great. Okay.
Fantastic.
Thank you very much for having me today. Thanks to the Statewide Biobank for the invitation. Just before I dive in, I just wanted to say that this is an adaptation of a workshop that was given in ISBA in Melbourne earlier this year, where I presented alongside Mariana Bledsoe and Marianne Henderson. So let's dive in. So as many of you will know, the journey to publishing biobanking research, or indeed any research, is rarely linear. So I thought we could play a game of Publishopoly today. So this is a game that all aspiring publishers will be familiar with. Of course, the ultimate goal is to pass go, collect $200 and have your name appear in print. So let's dive into this and break it down a little bit more. So starting with the acquisition of data and combining this with your own ideas and imagination, we then proceed to writing the manuscript, selecting a journal where your work might fit, and then submitting your final draft with a cover letter. And from here, your journey can take a number of turns. So the journal editor can reject it outright. Hopefully that's not the case. If it is, it might send you back to the submission process at a minimum, and quite possibly sending you back to the writing and journal selection aspects. They can also choose to circulate it to a small number of selected reviewers for peer review. Again, here your fortune's in the hands of the peer reviewers, who can recommend in concert with the editor minor revisions, major revisions, or again, rejection. But if you can respond to these reviewer comments well, then you're well on your way to passing go and acceptance of your manuscript for publication. So let's dive in with the first aspect, which is planning. So the first question to ask here is whether the data and ideas have been previously published. And this obviously requires an awareness of the existing literature. It's pretty good practice to also do a literature search just to double check. And related to that, you just need to consider what's new or original in your planned work. Is there some point of difference to the body of literature that's already out there? So once you've got those points down pat, it's time to consider your writing and research team. Depending on the size of the team and also the extent of your existing working relationship, you might want to consider either a formal or an informal agreement. So even at this early stage, it's worthwhile thinking about how to maximise your research impact. So what I've put here, or I'll run through, is some ideas that a blogger from Nature came up with of highly cited papers. So the first is a shortish title, so around 10 words. Indeed, I think the blogger said the sweet spot was between 7 and 13 words. Also with the title, where possible, make it something that reflects current topics of interest or methods. Larger authorship teams were also linked with a high number of citations. This might be because you've got more of a multidisciplinary team or more of an international team. And then also longer length papers. So over 5,000 words were more highly cited. And finally, within the journal's limits, of course, the more figures and tables, the better. So the blog actually suggested a minimum of 6 figures and 2 tables. So turning now to drafting, there's, of course, many different types of articles that you could be trying to draft up. There's a great framework overarching that's developed by the International Committee of Medical Journal Editors, or the ICMJE. And this is called Recommendations for the Conduct, Reporting, Editing and Publication of Scholarly Work in Medical Journals. So in particular, there's a section there that I've put, which is called Preparing a Manuscript for Submission to a Medical Journal. And there's quite a bit of valuable information in there. So I'll come to journal selection, but just to note at this stage that following author guidelines for your selected journal closely is always a good idea. Finally, I just wanted to touch on AI. I speak to my colleagues about AI a lot and I get really mixed responses. Some people love it, some people hate it, but I think we can all agree that it's being used increasingly as a writing tool for researchers, and it's got a spectrum of uses, everything from initial drafting through to just using it as a revision or a finessing tool. So it's important to note that AI promises faster and more polished writing, but there are human elements to writing academically that are quite challenging for AI to emulate. So even in academic writing, there's this element of emotion, some stance or position that must be taken to generate a persuasive argument. And that can be difficult for bots to get their head around. So the main things with writing academically are flow and accuracy, which AI can do to some extent. But the main issue is depth. So you can find that AI-generated writing has this sort of mechanical perfection to it. And this is just a few pointers that I've put together to keep in mind during the drafting process. So firstly, logic and flow are key to keeping the reader engaged and following the narrative. Conversely, a lack of focus and narrative will diminish readability, even if your data is excellent. Methods should include enough detail to enable reproducibility of your findings. Conclusions tie your story together, and they can take into account things bigger than just what you're trying to present in your papers. So you can look at things like scalability or applicability in other settings, policy implications, what the findings mean for biobanking in general. Next, figures and tables should be really clear and concise and obviously should follow the journal requirements. Grammar and sentence construction is a big one. I've put some resources at the end of this presentation and I think it's going to be shared up online. And citing. So others' material must obviously be referenced and explained appropriately within the text. And in general, also avoid old references. So if we turn now to considering journal selection, so when we think about research involving biopreservation, biobanking, there's this huge scope of topic areas. and obviously for both human tissue and non-human collections as well. And therefore, different topics are going to suit different journals. So in general, there's a couple of factors to take into consideration. Firstly, that you'll want to marry your intended audience with the readership of the particular journal. So for this, you can look at the journal's aims and scope on their webpage. So their webpage will usually have a brief introduction to the journal, an outline of subjects covered, and the types of articles published and not published as well. Secondly, along with that, you, as a general rule of thumb, the higher the journal prestige, the harder it is to get published there, unfortunately. So there's a few different journal level metrics. They all revolve around the number of citations received in a defined time period. So we've got the journal impact factor, Simago journal rank, and the site score, which is a Scopus product. So those 2 points need to be considered simultaneously. So you might have a case where your paper might be reviewing biobanking practices in a particular country or region, and that might be better placed within a journal in that country rather than an international journal. Or you might have a case where your paper's discussing a novel cryopreservation technique, and that might be better placed in a specific biobanking journal in order to reach the right audience, no matter what the journal impact factor is. And finally, I've just got a point there about costs. So it's worthwhile considering, even at this journal selection stage, are there costs involved in publishing your article? And if so, do you have any funds to actually cover that? So even with all of that in mind, it can be like looking for a needle in a haystack until you get to know journals in your area of expertise. That usually comes from reading widely, and obviously you can register for Google Scholar or PubMed keywords to help you receive targeted lists of relevant papers. You can discuss journal selection with your peers, even if they're not listed authors. And another option is to write to the editor-in-chief of a journal to pitch your idea. But bear in mind, it could go either way. They could reject you outright without ever seeing what you've had to say, or they could just direct you back to the submissions process. So I've just taken a screenshot here of an online tool. It's called Jane, so the Journal Author Name Estimator. So online you can enter the title of your abstract, title of your manuscript. You can even put the whole abstract into a text box. And then Jane will formulate a ranking of the most relevant journals based on the information that you've inputted. So for this one, I simply typed in biobanking into Jane as a keyword, and it's a bit small, but perhaps unsurprisingly, the top journal identified was Biopreservation and Biobanking. So at least we know that Jane works. And for this graph, I just performed a very quick Scopus search of papers that included biobank, biorepository, biobanking in the title, abstract, or keyword. So here we can see the top 10 journals in the last 5 years. Just some notes on manuscript submission. It's generally via a web portal. I always find it takes longer than expected. For each author, you might need one or more of an ORCID, which is a 16-digit identifier for researchers, a Ringgold location, which again is an identifier for academic institutions, and or a signed contribution statement of which aspects of the manuscript development that each author was responsible for. So as you can imagine, if that authorship team is on the large side, it can be a pretty onerous task in and of itself. It's worth also noting that all named authors are expected to take responsibility and accountability for the submitted work. And finally, proofreading is absolute key here. So there's a whole heap of details to get right at that submission stage, and it always takes me more than one go to get everything in the right order so that when the generated PDF comes out that it looks right. Just things like file sizes, file types, and so on also can take up quite a bit of time. So I'm going to turn here to one aspect of the submission, which is the development of a cover letter. So some people think that cover letters are antiquated. Some journals state that they're optional, but they do provide a good opportunity to sell your manuscript idea to the journal editor. So it's an opportunity to provide information on the importance, the relevance, the originality, the defining outcomes of your manuscript, and it provides a hook to the editor on why they should take a further look. It might also help to prevent desk rejection straight back by the editor, as we looked at in that publishopoly flow. And you can also use that as an opportunity to provide other information there, like reviewer suggestions. So this is an example of a cover letter from my own work. So in green, we've got important features. We've got an institutional letterhead addressed personally to the editor-in-chief, dated. The first paragraph introduces the type of article, the name of the article, and the name of the journal. And in this second paragraph, when we get into selling the reasons why the article will be of interest to the journal readership, and justifying why the journal should consider publishing the work. The second page just has a statement that the work is original and hasn't been submitted elsewhere for publication. Thanks the editor for considering, provides some contact details for the corresponding author, and as I said, lets the editor know any other relevant details. So just some do's and don'ts for cover letters. Do include a cover letter, even if it's not mandatory. Be polite and professional. Proofread your cover letter. It's the first thing that the editor will probably read. Declare any conflicts of interest. Be succinct. And you're within your rights to include names of anyone you don't want to review your work. Don't make it too long. Don't just repeat what's written in your abstract. And don't refer to your reputation in the field or how many papers you've already published or what they are. So as referred to earlier, from here on in, there's a few different ways that your submission is heading. So the first of these is the best outcome of all, which is acceptance. So what to do here, relax in the knowledge you'll soon be a published author, think about your next big paper, but bear in mind it's probably pretty rare for this to happen first up. So the second one is the exact opposite of that, which is outright rejection. Here you should take some time to feel bad and then take stock of your options. So in reality, there's three options. You can submit to another journal, so you can look around for journals that are a better fit, lower prestige, a combination of those. Secondly, you can go back to the drawing board. How could you improve the methods or the writing or the implications or reconsider them? And the third option is really to move on to something else and perhaps reconsider this paper at a later date. Our next option is major revision. So there's work here to be done. The news is generally good though, so take a deep breath, start work on the revisions and put together a clear response. And then minor revision is even better than that. So almost all papers will come back with some sort of changes to be made. So you should rejoice in the fact that yours are minor and put your mind to making it as quick and easy for reviewers to see the changes you've made and accept your paper. And just got some arrows here to indicate that major and minor revisions, we really want to turn them back into an acceptance. So just a case study of a recently accepted paper of our own. This was an original manuscript. There were 3 reviewers and there were 33 comments or queries to respond to. So again, the usual in green, and the main thing here is that we describe our method for responding. So my eyesight's really bad, but I think we've said reviewers' queries and comments are shown in italics and the responses are in ordinary font, and any new text is shown in red, and all page numbers refer to the revised manuscript. And then we list them out. So here we've got the reviewer's comment in italics, our response. It's an interesting point. We've added it to the discussion. We've identified the page number and we've put the new text in red. And conversely, then we have a reviewer comment that we respectfully disagreed with. So they've said the graph requires considerable reorganisation. And we've responded saying we've carefully considered your comments. provided some justification, and then therefore we've retained the figure in its current state. Again, just a quick do's and don'ts. So do respond to every query and comment from reviewers. Thank and acknowledge their efforts, bearing in mind that most of reviewers are working for free. Explain your reasons for any comments you disagree with. Mark up your changes in the manuscript and refer to where in the manuscript the changes have been made, either a line or a paragraph or a page number or all of the above. Don't be rude or disrespectful. Don't make it difficult for the reviewer to find your changes that you've made, and don't ignore comments that you don't want to address or that you don't think are worth responding to. Okay, so we're getting to the end, the best bit, the relief. You've been accepted. There's just a few considerations at this stage. Reviewing the final layout. So don't just assume that an exact copy of your text has been pasted straight into the final layout. There's often typos or misinterpretations of what you've meant or other errors to be found at this stage. It's worthwhile reading it word for word. You'll need to make a payment if there's any at this stage. So there could be open access fees or in the old days, colour printing fees, probably less relevant now. You might get a big shock if you're in Australia and you've seen the open access fees and missed the US dollars. Then once all that's done, the paper will usually be available online, but it might take some months for it to actually be assigned an issue number. And that's my resources, and that's it. So I'm going to stop sharing and hand over to my colleague, Beth. Thank you.
Hi, everyone. I'll just bring up my slides. Alrighty, hopefully everyone can see them now?
Yes, thanks.
Great. So continuing on from Amanda's awesome presentation, I'm going to go back to the beginning of the Publishopoly board and figure out, well, what if you don't know what to publish? And so this was me 2 years ago when I started at the Statewide Biobank. I thought, we have nothing to publish. We just process samples. But over the last few years, I've uncovered within our databases and within our internal knowledge, there's a wealth of things that we should be sharing with the broader community and should be publishing. So a few disclaimers, and don't worry, I'll get to why there's an ice cream scoop on the screen. But I'm very green in this area of publishing and biobanking. I've published one paper this year. And so I'm not an oracle of knowledge in this area, but I have a lot of ideas, and I'm hoping that you will scoop them from me and beat me to publication, or work with us at the Statewide Biobank to bring some of these ideas to fruition. So many of you on this call will be familiar with the biobanking pipeline, where a consented participant will donate a specimen that's transported to a processing laboratory. Some quality control measures might be immediately performed, or even some downstream analysis. But it's most likely that the specimens will be stored for a long period of time, with a small proportion of those being thawed and used for downstream analysis. Now, what we're very comfortable with analysing on a whole is when we're looking at these downstream results, how they differ within a patient or a participant cohort. So, for example, with a cohort of participants with and without cancer, it'd be very common to look at the expression of different genes, for example. But what we don't do all that well is look at, well, how does variability in the middle of this pipeline impact our results downstream? So this brings me to the first area of potential areas for publication, which is the impact of pre-analytical variables on biospecimen quality. So this will be looking at pre-analytical variables such as those coded in SPREC, the standard pre-analytical code. And many of us that have been collecting specimens for quite some time and have built databases based on ISPA and other biobanking best practices would have this information already. So this would detail like the type of specimen, the type of container that it was collected in, what temperature it was held at before processing and how long it was held, how it was centrifuged, and how it was stored. Detailed in SPREC that does create or can create variability is other things like how it was processed. So at the Statewide Biobank, we have some of these things that we do record that could create variability, which is, for example, how it was processed in terms of using automation or manually. So while we aim to do everything with an automated instrument, if the equipment is down, we have a manual technique, which could introduce variability later. There also could be different types of instruments that are potentially the same instrument, so we have different of the same one, or different types of the same purpose. For example, different brands of cell counters. And another example is different types of reagents. Maybe there is one cryopreservative that was used for a study and later was switched over to a different brand or type. So this is an example of build your own graph. So on the x-axis is this pre-analytical variables that I've listed here that you could delve into more to figure out if there are any differences on biospecimen quality. So now let's look at the y-axis. So what are measures of specimen quality? So for some of these would be internal quality control measures that are performed at the time of extraction. For peripheral blood mononuclear cells, this could be yield and viability. For serum and plasma, this would be the Hill index, and there's other metrics for nucleic acid. For other laboratories that do perform downstream assays, this is where you could also look at expression levels and functional assays and how these results vary with different pre-analytical variables. So now let's go into our published example. So earlier this year, I wanted to look at, we've done roughly 2,000 PBMC isolations over about a 1.5 year period. And these were performed using either a manual or automated protocol. And so I wanted to know, is there any difference between these two techniques? Additionally, as this study it collects specimens nationally and sends them to our laboratory, they could be up to 10 days old as per their study acceptance criteria. And as we all know, that specimen quality does deteriorate over time, but to what degree? Most of the papers I've seen only go out to 48 hours. So what do PBMCs look like after 10 days? So I looked into our data and I found some rather interesting results. So here we have on the left, cell viability of our PBMCs, and on the right is yield. And in the black bars is our manual process, and in the white is automated. And so we found there was no significant difference when we were comparing these two methods. And rather surprisingly, cell viability was over 70% for over samples that were over a week old. But what didn't surprise me all that much is that yield decreased over time, but the level of which it was interesting to see that samples up to 5 days old had a reasonable yield, and it did decrease as expected for samples up to 6 to 10 days old, but there were still viable cells at that endpoint. So this data is quite interesting to look at both internally, but now showing it externally so that others, when they're setting up their study design, they could think about, well, can they have a larger acceptance criteria if you only need a small number of cells? So depending on their downstream purpose. And this could inform things like if you were doing a large study, if you were to use a centralized or decentralized biobanking model. So benefits of looking at these pre-analytical variables will inform the study design and downstream application. It could additionally identify critical areas to control, or those that can be flexible. Looking at that data, we found that the automated and manual method had similar results, so we won't need to control that all that much. And additionally, this could reveal cost-saving initiatives. So like with the previous example, that could reveal if a centralized or decentralized biobanking model could be used, or could be other things. So for an example, if we were to look at within your data, if you could send samples at both room temperature and at 4 degrees, if you notice that downstream there is no difference when you send them at room temperature, that could save on shipping costs. So the next area I'll quickly go through are looking at stability studies. So as we all know, specimens over time in long-term storage will deteriorate in quality. So as a whole, we're very comfortable at analysing specimens long-term and how that will impact our downstream results. But what we don't do all that well is looking at the big picture. How do variables in between this whole pipeline impact specimens over time? So this is a published example that I'm sure we've seen very similar things over the years of looking at how a specimen looks over time. So here's a graph of PBMC cell count over a 12-month period in long-term storage. But what we could be looking at is how to pre-analytical variability impact specimens over time. So for example, if we compare our PBMC method, both automated and manual, over long-term storage, that could be something really interesting to go into. And so these are very similar benefits to the first area they talked about, but also ensuring that specimens have long-term usability. And so the final area is publishing methods. So our processing methods, there is a very limited publishing domain of processing methods with representative data. So with that example I showed earlier, that was a part of a paper where we looked at our PBMC isolation method. And so in that publication, we additionally put in a detailed method, as well as we had a video method. So this screen grab here is of me performing the method for the cameras. So that was an interesting experience. If anyone would like to hear more about that, happy to talk about it. So by publishing our methods, it can promote standardization and transparency in biobanking. But additionally, when we are writing our methods up and validating them, if we have the intention to publish, it could increase the data quality. Now, when we're looking at method development, we ask the team, is this publication ready? And it's likely that it does need more repeats and more, a bit more done. So at the end of the day, we can be rest assured that our methods are really well validated because we've had that publication mindset from the beginning. For anyone with ISO 9001 or 20387 certification, method validation is a requirement. But if you're looking to get certified, this is definitely a good place to start of detailing your methods and having representative data of high quality. And so with that, this is our 3 areas that I've talked about. So we could be publishing more on the impact of pre-analytical variables and biospecimen quality, looking at sample stability over time, as well as our methods. And so I encourage you all to look, if you've been collecting specimens for some time, to see if you have this data already, or what is the gap? What do you need to start collecting so that you could use this data downstream? And so I thank you all for listening to the presentation. Hopefully there were some exciting ideas there. Happy to collaborate or discuss more of these ideas in the future. And I'd like to thank our funders, the Office of Health and Medical Research, as well as our support from New South Wales Health Pathology, Sydney Local Health District, as well as the members of the Statewide Biobank, with special thanks to my co-authors, Maria, Sarah and Kiki, and Candice and Jenny. Thank you.
Thank you both. That was a great pair of presentations, I think really complementary. I'm sure that people have got questions or comments, so if anybody would like to ask anything, yeah, please hang on. I think there's something in the chat. Let's have a look. Yes, Karen's applauding, and Dan did have to leave early on, but his comment was good stuff, which I firmly endorse. So if anyone has any questions, please, yeah, put up your hand, write it into the chat. I guess I might start with just thinking of a question for you, Beth, around, like, I think you've suggested some topics of, where people might already have data. And I think that's a really important place to start, to think about, what you've already got. That's the quickest way, of course, to a publication. But I guess I'm wondering whether you could think about are there other things that you also think that biobanks could do with just a little bit of additional foresight? I think you've talked about, how your intention to publish can result in better quality data, which I think is a fantastic point. But are there other topics that maybe with a little bit of extra effort, you might be able to think about putting something together? I guess one thing that I really like reading in biobanking, narrative descriptions of how biobanks work, and there's actually not a lot of those in the literature. Just talking about how your biobank operates, I think, can be valuable if there's messages in there that are generalizable, I guess.
Yeah, there's even more like commentaries of experiences about things that we don't talk about all that much. And even like letters to the editor based on publications that are out there, bringing in your own experience. So you don't need raw data to do that. It's just more bringing together what you have experienced. I think we're good about talking to our collaborators about these sort of things, but putting it on paper is something that I think we need to do more of.
And I guess there aren't really, I mean, Amanda, you mentioned that your presentation was also given at ISBA along with other editors from Biopreservation and Biobanking, but I'm not really aware of any, there's not really a lot of like courses or hands-on sort of, or workshops in terms of writing for biobanking. I've never seen any of those organised. Have you ever seen anything like that at an ISBA conference, Amanda?
I haven't. I guess in terms of a Venn diagram, there's overlap between writing in general for journal articles and biobanking research, which is more what Beth has gotten into. There's definitely a lot of resources online for the former, but I agree not so much for the latter. I also just placed in the chat, my understanding is that in more recent years, the journal Biopreservation and Biobanking has steered away from the descriptions of biobanking activities that you were referring to, Jenny. But this journal that I've put in the chat, I think that's more their bread and butter, is to highlight that kind of thing. So just in case people weren't aware of that one as well as another option.
Yeah, that's a good point. I guess, yeah, I think that the Biobanking Journal has sort of shied away from the articles that I've sort of described as here is my biobank kind of articles.
Yeah.
But I think sometimes, it's not so much an individual biobank, but more, maybe combined experiences in a region. I recently read a paper in I think it's the BMC Journal of Medical Ethics or something like that. There's definitely some of those more narrative papers that can be published in ethics journals where people talk about, some of the challenges that biobanks experience in particular settings or working with particular kinds of patient groups and things like that, and ways that they've, managed those kinds of issues. So I quite like reading those because I find them, they're papers that talk to you in quite a real way. So I suppose,
Yeah, look, I think it's as close to you get. It's a formalized way of, networking at a conference, and it's probably as close as you get to that. And as we know, there's a lot of wheel reinvention in biobanking, and this is a way of sharing that sometimes informal knowledge that can be of value to either new setups or also for established ones.
In terms of the question about like the letter to editor, I'm aware of discussions around that. Some people kind of think, oh, well, we don't need letters to the editor anymore. But I think certainly many journals would really expect them. And if they don't see them, they view that as a bit of a kind of a red flag. Is that also how Biopreservation and Biobanking sees it? Do you mostly see letters to the editor coming in, or?
I actually don't know. I suppose only the editor is privy to that. It certainly doesn't come with the documents for review. Yeah, so I don't know the answer to that. I think the editor in that particular journal is very established in the field and probably is aware of not the majority of papers, but some papers that are coming through before, like through verbal conversations So that's another avenue that's a bit less formal than a cover letter, but yeah.
Yeah, and I guess that's a good point too. Like some authors actually make pre-submission inquiries to journals. Yeah.
Yeah, that's right. So actually, just last week, I made a pre-submission inquiry, and I have done in the past as well, and just been told we don't accept this information either for capacity reasons, or maybe they feel that it gives some sort of unfair advantage or promise of an outcome. And so then, as I indicated in my talk, you can just circle back to the submission process anyway.
Yeah, that's true. That's true. And Beth, when you were working with the Journal of Visualized Experimentation, I mean, did you make a pre-submission inquiry to that journal? How did that connection happen?
Yeah, I reached out to the editors and just said, are you interested in this type of thing? Like, I haven't noticed any biobanking methods on your, journal. Would you be interested? And they said yes, and so gave me a invitation to submit. So I had a formal link going. It gave us a time crunch though, because we just had the idea. So they wanted a paper now. It's like, oh, we have to do the work now. So that's the only thing, the caveat of that. You have to be ready to start if you do that.
Yeah, that's a good point. Yeah, that's a good point. And so I think your paper's online, and the video, we're just waiting for that to be put online as well.
Yeah, it should be next week. We've done the final proofs.
Yeah, do you want to put the link into the chat?
Sure.
Yeah. Does anyone else have any other questions? I think we've got certainly some experts here that can help. I've certainly, I really like the publishopoly analogy. It is a bit of or snakes and ladders is the other thing. But I think there are certainly some things that we can all do as authors to make that pathway easier. And, some of the advice, like always being respectful, recognizing that your reviewers have given their time to consider your work, and, giving thorough responses to queries is always helpful. So sometimes just putting a little bit more time into things in the short term can generate savings of time in the long run. So we've got a question from Albert. Should journals accepting publishing in the biobanking field request more hypothesis-driven articles? Yeah, that's a good question, Albert. I don't know, Beth, what do you want to answer that? Both of you.
Yeah, it's an interesting question, because it's, depending on which part of the biobanking pipeline you're looking at. So if, it's looking at expressions of proteins in cancer populations, then yes. But sometimes best practices and experience will dictate how to set up a study. So having that data and those experience are really beneficial to have in the domain. So I think we need to have a combination of the both. But I'm sure Amanda, being on the other side of it, could have some thoughts too.
Yeah, I think the obvious answer is that a balance is best. I think perhaps more of the scientific journal articles might actually end up in journals that biobankers aren't looking at, so they might actually be lost out there, and we haven't identified them for that reason. So it's a bit difficult to actually understand the landscape of all biobanking literature, but you can see from the graphs that I put up, or the Jane tool, that in general, if you're just looking for anything to do with biobanking, then that biopreservation and biobanking seems to win out there.
And Amanda, are there particular, like in terms of instructions to authors, are you aware of whether biopreservation and biobanking does request things like manuscripts to state hypotheses or aims? Are there categories of manuscripts where that's an expectation, or is that something that gets discussed amongst the editors?
No, not so much. I think they're more concerned about the higher level things and the instructions to the authors. So I think for aspiring publishers, there's that combination of what's happening on the journal level page, but also at that ICMJE level.
Yeah, thank you. Well, if we don't have other questions, please join me in thanking our speakers. Thank you so much. That was really a great talk. I'm sure a lot of people are going to benefit from the recording as well. And I'd just like to thank everyone who's joined our seminars this year. They're a great pleasure to host. I think we learn a lot, and we hope that people who attend learn some new things as well. And if you do have any ideas for speakers for next year, we'll certainly be back in 2025. Please send me an email, or you can just email the Statewide Biobank. We'd love to hear from you. But thank you again very much, Beth and Amanda. Fantastic talk. Great way to end the year. Thank you. Bye.